Okay, moving on from carbohydrates to fats or lipids. Um, lipids have, in some ways, a simpler structure. They tend to be mostly carbon and hydrogen, as you can see from the fatty acid, palmitic acid here at the top of the shaded region. Nothing but a long chain of carbons saturated with hydrogen atoms, which is why it's called a saturated fatty acid, meaning every carbon is bonded with as many hydrogen atoms as it possibly can to be a part of a chain like this, with the exception of the carboxyl end of the molecule, which is why it's an acid. Remember from the last video we talked about the carboxyl group, one of those functional groups, being a part of an organic molecule that would cause it to be an acid. And there is that carboxyl group at the end of palmitic acid, and down with the unsaturated linoleic acid, there is a carboxyl group at the end of that as well. The unsaturated fatty acids have bends and kinks in the chain, and because of that, that bending causes the fatty acids to have a much lower melting point, and so they tend to exist as liquids at room temperature and body temperature. And those are the more healthy types of oils and fats that mainly come from plants, and animals tend to make lots of saturated fats like this, and those are not so healthy for our bloodstream, and that's why we're urged to avoid too much saturated fat in our diet. This is showing one of the more stupid human creations here, and also demonstrating some important chemical terms, cis and trans. Cis means same and trans means across, and so we are looking at the double bond in these two fatty acids, oleic and eleic. I don't know how to say that. I know oleic though. The trans fatty acid has a carbon-carbon double bond in the chain, and then hydrogen atoms on opposite sides of the chain from each other, and that's the across or trans form of that kind of double bond. The cis double bond has the two hydrogen atoms on the same side of the molecule, and where it's a cis double bond, and this is the sort of naturally occurring kind of double bond we see in fatty acids, it causes the fatty acid chain to have a kink, a bit of a hairpin turn there. And that's what will give these types of fatty acids, the unsaturated versions, this is what gives them their lower melting points and their more healthy characteristics. We took healthy fats like oleic, and we found ways to artificially make them unhealthy fats, because that way we could make things like margarine and types of fats for cooking where those would be solid at room temperature, and we created something that's actually as unhealthy, if not more unhealthy, than the animal fats that we were trying to substitute for. Nice job. Animals store their fats often as triglycerides. This is the way we store energy long-term in our body. If we have excess energy at the end of the day, we'll ship some of that energy in the form of precursor molecules to fat cells, and they will build these triglycerides, these fats. And a triglyceride is composed of 3 fatty acids, like the ones that we just looked at. And this is showing a dotted line here representing that this chain could be significantly longer, and it typically would be for the fatty acids we would build our fats with. And then we would take those 3 fatty acids, combine them with a glycerol molecule, which is 3 carbons long. So at each one of the carbons, we would connect one of the fatty acids, and now we have this complex fat called a triglyceride. And this is one of those dehydration synthesis reactions, only it's a triple dehydration synthesis to bond these three fatty acid chains to the single glycerol. There are the water molecules that come off from it. That's how we build our fats. A couple of other important fats to talk about before we move to the next category of organic molecules. Phospholipids are sort of similar to the triglycerides, but they don't have three fatty acids bonded together in a glycerol. They have two fatty acids, and then instead of a third fatty acid on the third carbon of that glycerol, we have a phosphate group and often some other atoms bonded to that. In this case, it's a choline group, which has a nitrogen atom with a charge on it. And the negative charge on the phosphate group, the positive charge on this choline group, causes this part of the molecule to be quite polar. Those opposite charges would attract the positive and negative sides of water molecules. And so we would say that this end of the molecule is hydrophilic, water-loving, whereas the hydrocarbon chains that make up the fatty acids, and those are simplified by the zigzag lines here, those tails of the phospholipid are nonpolar. There are no polar bonds between all those carbons and hydrogens, so there's no positive negative ends for water molecules to be attracted to. And so we would say that that's a hydrophobic or water-hating region of the molecule. And when we put phospholipids in water, they behave in a very interesting way. They will orient themselves so that the water-hating, water-fearing hydrophobic tails will all orient towards one another, and the hydrophilic phosphate-containing heads of the molecules will orient themselves outwardly towards the water molecules. And if you remember from your intro biology classes, biological membranes are double layers of phospholipids, and they arrange themselves in those double layers so that the hydrophilic heads of the molecules are facing the cytoplasm inside the cell and the watery medium outside the cell. And the hydrophobic tails are facing each other in that double layer. And we'll see some diagrams of that coming up pretty soon. The last category of fats to talk about, and we'll just do this very briefly, is cholesterol. It says on the slide there that cholesterol gets a lot of bad press. It does. But in fact, we make cholesterol in our body. It is an essential nutrient, it is an essential molecule, because cholesterol is the starting point for making a whole slew of different steroid hormones. And three of those hormones are shown on this slide: cortisol, testosterone, and estradiol. There are others. And so cholesterol is an important nutrient. We will actively absorb it from our digestive tract, and we synthesize it in our cells as well. But too much cholesterol is not particularly good, especially too much cholesterol bonded to the proteins that tend to transport cholesterol in our blood. Those, like the saturated fats that we talked about earlier, not real good for nice smooth flow through our bloodstream. Those saturated fats and cholesterol can form deposits on the walls of arteries, and those deposits can restrict the flow of blood and potentially become so big that they cut off the flow of blood. If that happens in a blood vessel that's feeding part of your brain or your heart, you will have a stroke if it's your brain, a heart attack if it's your heart. So it's bad news. So proteins are hugely important. And all proteins are made of amino acids. And we mentioned those functional groups in the last video. Each amino acid has an amino group on one side, a carboxyl group or acid group on the other side, and then amino acids differ by the R group, as it's known, that's attached to that central carbon atom. And the R groups can be very different from one another. They can be small and simple. They can be large and complex. They can be ionic. They can be acidic. They can be basic. They can be hydrophobic, hydrophilic. And so depending on what is sticking off this R region of the amino acid, that amino acid might have a very different chemical character. Proteins are chains of amino acids, and like the other chains that we talked about, we put together these individual subunits by a dehydration synthesis reaction. They call this a peptide bond, and well, it's because it's making peptides. Those are proteins, but really it's dehydration synthesis, not any different than the other types that we've seen before, but it gets a special name. And then those chains of amino acids, which is just sort of a very simple structure, a long chain, doesn't stay simple. Because of the different chemical characteristics in the 20 different amino acids that we use to build our proteins, the amino acid chains can fold and coil and twist and fold back over themselves into extremely complex shapes. So we talk about the structure of proteins as having different levels. The primary structure of a protein is just which amino acid comes first and second and third and fourth in the chain. So it is, the primary structure is the sequence of the amino acids in a particular protein. And then those chains of amino acids then very often will do two things in localized regions of the amino acid chain. One thing that happens commonly is the chain will start to coil up like a spring, and that's called an alpha helix. And another thing that can happen is that adjacent parts of the amino acid chain can interact with each other and cause this sort of folding back and forth, and that's called a pleated sheet, like a pleated skirt, these folds that are coming in regular sort of arrangements. Both of those are said to be secondary structures of proteins. And then tertiary structure is how those coils and those folds all interact to make the final shape of the polypeptide chain. And that's tertiary structure. And then some proteins like hemoglobin, which is depicted in the slide here, hemoglobin has quaternary structure. Proteins with quaternary structure have more than one amino acid chain bonding to each other to make the final functional protein. In the case of hemoglobin here, there are 4 individual polypeptides. That's a protein. And those 4 polypeptides interact and bond with each other to form the finished functional hemoglobin that carries oxygen around in our blood. The structural diversity of proteins, the almost infinite number of different ways that you can twist and fold and coil and shape a protein, leads to an almost infinite number of different functions that are possible for proteins. And some of those are listed out there for you. And we'll run into all of those later on when we talk about different systems. Finally, we want to wrap up with the 4th category of organic molecules. That's nucleic acids. And I'm just going to assume that all of this is review from your biology classes. And if it's not, then I'm going to ask you guys to let me know about that, and we'll spend some time going over this next time in class. But I'm just going to let the slides do the talking for me, because I think you've heard enough of me. And read the slides, check over to see if this is ringing a bell from your studies in biology, and if not, let me know. This is showing the complementary base pairing across the double helix in DNA, and all of those are hydrogen bonds. That's what holds the 2 strands of DNA together. There's a simplified version of the twisted ladder, the double helix shape of the DNA. And here's some details showing that complementary base pairing and the hydrogen bonding that holds the two sides of the DNA together. The other important type of nucleic acid is RNA, and the names are different because of the different sugar components. It's either deoxyribose or ribose. Deoxyribose in DNA, you can see at this position of the molecule there is just a hydrogen atom, whereas in ribose there is an oxygen with a hydrogen atom bonded to it. So it is one oxygen less, deoxyribose. And then instead of having A, T, G, and C as the four nitrogenous bases in the chain of DNA, RNA has four different nucleotides, but instead of having thymine, they have uracil. So it's A, G, C, and U instead of A, G, C, and T for DNA. But you can see that thymine and uracil also very similar. And that's it. You're kidding. We must be done.